Hello everyone. In the video, we will talk about the structure of nucleus and how the chromosomes are present. What is the difference between chromosome, chromatin, centromere, centrosomes? All these terminologies we are going to discuss in this video. First, we'll see the structure of nucleus. Okay. From first, where the nucleus is present. The nucleus is present in all type of eukaryotic cells. Eukaryotic cells are of two types. One is animal cell, another one is plant cells. Okay. So here, if you take a normal typical cell, in the middle part of the cell, the structure first we have drawn is the nuclear membrane. Nuclear membrane. And ma'am, what is the gap? Is it important? Yes. These gaps are called nuclear pore. These gaps are called nuclear pore. And the space which is present inside the nucleus and the space which is present outside the nucleus is called by different terms. Yes. The space inside the nucleus is called by the term called nucleoplasm. It's called by the term called nucleoplasm, and the space which is present outside the nucleus is called by the term called, yes, I hope you know that, cytoplasm. Yes. The plasma which is present in the cell is called cytoplasm, and the plasma which is present inside the nucleus is called nucleoplasm. Okay. Now, in the nucleus we have some point of accumulation. Okay, a pointed structure. This is called nucleolus. What is the function of nucleolus? It acts as an active site for the production of a ribosome and RNA. Ribosomes and RNA are produced from the site called nucleolus. It is present inside the nucleus in some corner of the nucleoplasm. Okay, the main part of nucleoplasm contains what? The main part of nucleoplasm contains a thread-like structure called, yes, chromatin. It is called chromatin. Now we'll move to the topic called chromatin. What is chromatin and what exactly it is made up of? So chromatin is a thread-like structure. Okay, the length of DNA is 2.2 meters, but the size of the cell is less than that. So how the DNA is coiled and present inside the cell is in the form of chromatin. So chromatin is nothing but it's a coiling of DNA. So if we see from the microscope, the structure of chromatin will be like this with some beaded structure here. This yellow color one is called nucleosomes. This yellow colored one is called nucleosomes. See, there are different terminologies. One is nucleus, another one is nucleoplasm, another one is nucleolus. This is another one terminology which is called nucleosomes. Okay. Now, if we zoom the nucleosome, what this nucleosome is actually made up of? It is made up of a protein called histone, H2A, H2B, H3, H4. What are these? These are proteins, okay? H2A, H2B, H4. Okay. Now these proteins are present at backside also, like this. In three-dimensional view, we can view this properly. So backside. So now totally 4 plus 4, we have 8 proteins. It is called histone. What is histone? Protein. The structure is called histone octamer. histone octamer. What is histone octamer? It is made up of 8 proteins of H2A, H2B, H3, H4. Someone is missing here, right? Where is the H1? Yes, H1 will be present here. The H1 will link the chromatin, the DNA, the green colored one which I represented here is nothing but it's DNA. Is there any different terminology between that DNA and this DNA? Yes, this DNA is called core DNA and this is called linker DNA. This is called linker DNA. Okay, now what is DNA? What is DNA? DNA is nothing but deoxyribonucleic acid. Deoxyribonucleic acid. Okay, what is this deoxyribo? It is a sugar. It's what? Sugar. Okay, now the DNA is made up of nucleotides. Okay. Now your next question I can guess, what is nucleotide? Yes, nucleotide is nothing but we can simply remember by the formula. What is nucleotide? Nucleotide easy by the simple formula. Nucleotide is the combination of sugar plus phosphate plus nitrogen base. This is called nucleotide. Sugar plus phosphate plus nitrogen base. The sugar could be of two. One is ribose, another one is deoxyribose. And is there any similarity between these two sugars? These are sugars. Is there any similarity? Yes. These both are 5-carbon sugars. We can simply call it as pentose sugar. We can call it as pentose sugar. Okay. Now we understood the DNA is made up of nucleotides and the nucleotides can be simply broken down into sugar plus phosphate plus nitrogen base. If you have doubt what is this nitrogen base, there are 4 types of nitrogen bases which is present in DNA. 1 adenine, 2nd thymine, 3rd cytosine and 4th one is guanine. Adenine, thymine, cytosine, guanine. Okay, these are all the 4 nitrogen bases. Okay, these are all the 4 nitrogen bases which will be present in the shuffled form and it makes up the entire molecule of DNA. Okay. Next, what is the difference between DNA and gene? What is the difference between DNA and gene? Genes are present inside the DNA. Genes are nothing but a sequence of nucleotide. Genes are nothing but a sequence of nucleotide which codes for RNA, or we can simply say it as which codes for proteins. Okay. So genes are present on DNA and the DNA makes up the chromosomes. So here first we understood the structure called, this is chromatin. Now the chromatin will condense to form a structure called chromosome. A structure called chromosome during cell division. Yes, during cell division, the structure called chromatin will condense to form a structure called chromosome, and this chromosomes will have the genetic material called DNA. And the center part, which is present in the mid of the chromosome, sometimes not almost, and sometimes it can be present at different positions, different locations. Thus, the center, that the circle part is called centromere, which connects the two arms of the chromosomes, and around the centromere we have a structure. We have a structure called kinetochore. This kinetochore acts like a handle. Have you seen the handle which is present in the doors of your home? So the handle helps to move or pull or push the door. The same way, these chromosomes will be pulled apart during the cell division with the help of structure called kinetochore. To the kinetochores, some special structures attached. Spindle fibers. These structures are called what? Spindle fibers. These structures are called what? Spindle fibers. Okay. Now, if we are talking about cell division, we can talk about the duplication of chromosomes. So, if we take one chromosome before cell division, before any cell divides, the number of chromosome will be same, 46. This is chromosome number 1. Like this, we have 45 other chromosomes. So, together, totally it is called 46 chromosomes. It's before cell division. During cell division, what will happen? During cell division, the amount of DNA will double. So, DNA will replicate or duplicate. Okay, replication of DNA will happen so that the amount of DNA which is present in the chromosome, the amount of DNA, the point you have to remember is the chromosome number will never double, only the amount of DNA will double. Okay, and here we are going to learn the importance of this structure called centromere. What is the importance of centromere? Is the number of chromosomes are counted according to the number of centromeres. For example, you take this, you can consider this as a chromosome number 3. We have 46 chromosomes, right? It is present in 23 pairs, so you can consider this as a chromosome number 3. Ma'am, why not 4, 5, 6? It's up to you. You can select randomly any one number and you can keep any numbers, okay? So, I just took randomly chromosome number 3. So, what will happen to chromosome number 3 during the cell cycle and cell division? The amount of DNA is going to get doubled. Like how? Like this. So, the yellow colored material which I am drawing here is the DNA. So, here you can clearly visualize that the amount of DNA gets doubled. Now, the structure or this arm, this extension is called chromatid. I hope you understand the difference between chromatin and chromatid. Chromatid is the extension of the DNA. Here, in this chromosome, we have one chromatid for one chromosome. Totally, how many chromosomes we do have? Sorry, it's chromatid. It's chromatid. Each chromosome, if possessing each one chromatid, then 46 chromosomes are going to have 46 chromatid. But here you can see the chromatids get doubled. So we can call these two chromatids as, any guess? Yes, it is sister chromatid. Sister chromatids, and here we can count the chromosome number. The same chromosome number 3 here we have taken, it got doubled. Okay. So the total number of chromosomes, the chromosome number will be counted with the number of centromere. So the centromere is going to be the same, so the number of chromosomes are going to be the same, 46, but the number of chromatid will get double. So doubling of chromatid you can easily write, it's 92. Now the chromosome structure gets changed from single stranded to double stranded. During cell division, what will happen? The kinetochore structure is used to pull apart the chromosomes. These structures are called spindle fibers. During the cell division, what will happen? This chromosome will be cut into two equal halves like this. So the cell, the new cell which is going to get produced will receive equal number of chromosomes. Like the new daughter cell will get 46 chromosomes in each of it. Okay. So this is how the cell division takes place, and it is with respect to the number of chromosomes only. Okay. So here we can consolidate. Chromatin condenses to form structure called chromosome. So chromatin will be like this and the chromosome will be like this. Each cell we have 46 chromosomes, and out of 46 chromosomes, 23 chromosomes we are inheriting from our mother and 23 from father. So today we discussed about the nucleus and all the associated terminologies of chromosomes. In the next video, we'll see any other different topics. If you feel this video is very useful to you, share with your friends. Thanks for watching.